Hello friends, welcome back to my YouTube channel Nesting Padasali. So currently we are revising for a DBT BITP examination. So this is a past year question paper. So please read the disclaimer that I have given in the description below of this video. First question is, which one of the following antibiotic attaches to the 50S ribosome and inhibit the peptidyl transferase activity? The correct answer is chloramphenicol. So erythromycin, chloramphenicol, clindamycin, lincomycin are called as 50S inhibitor because they will be stopping the process of translation by attaching to the 50S subunit of ribosome. Whereas the tetracycline, spectinomycin, streptomycin, gentamicin, kanamycin, amikacin, nitrofuran, they are called as 30S inhibitor because they will be stopping the protein synthesis by attaching to 30S subunit. Whereas puromycin, mupirocin, they are called as inhibiting the process of translation, but they will be binding to transfer RNA. This actinomycin, rifampicin, they will be stopping the process of transcription, and this nalidic acid, ciprofloxacin, ovobiocin, which are called as quinolins, they will be inhibiting the process of DNA replication and other replication by binding to the enzyme called DNA guidase. The vancomycin, bacitracin, penicillin, cephalosporin, they are called as cell synthesis inhibitor, and the trimethoprim and sulfonamide, they are called as folic acid metabolism inhibitor. They are called as antifolate drugs, and this polymyxin and daptomycin, they will be disturbing the structure of cytoplasmic membrane. Next question, which one of the following is correct statement for active sodium potassium ATPases? Okay, so this particular sodium potassium pump, it will be pumping out 3 Na ion from the cytoplasm of cell to the extracellular environment, and it will be pumping out 2 potassium ion from extracellular environment to inner side of the cell. Okay, because if you take the structure of the sodium potassium pump, it has 3 binding site for sodium and it has 2 binding site for potassium. So it will be moving the sodium ion from the inner environment of the cell, that is from the cytoplasm, to the extracellular fluid, and it will be incorporating the 2 potassium ion from the outside of the cell to the inside of the cell, and for which ATP will be supplied. Okay, so Na plus K plus ATPase pump maintains a gradient of high concentration of sodium extracellularly. Okay, and it also maintains a high level of potassium intracellularly. So this particular concentration gradient is required for all kind of physiological process, like it is required for stabilizing your resting membrane potential, it is involved for regulating the cell volume, at the same time it is responsible for signal transduction of cell. Which one of the following genes are constitutively expressed and control the plant-induced activation of other Vir genes? The correct answer is Vir A and Vir G. Both are having a constitutive expression. This Vir gene will be activating a other Vir gene. So Vir A is a phenol sensing receptor which will be sensing the acetylstringent, a phenolic compound produced by the wounded plant, and then it will be first it will phosphorylate, autophosphorylate, and it will be transphosphorylate Vir G gene. The gene for 16S and 28S ribosomal RNA transcriber, it is transcribed by RNA polymerase 1. This RNA polymerase 2 is responsible for transcribing your messenger RNA, and RNA polymerase 3 is responsible for transcribing your transfer RNA and 5S ribosomal RNA. Next, the 2 Vir protein with nuclear localization signal which help in movement of tDNA to plant nucleus. So it includes Vir D2 and Vir E2. Both will be having a plant nuclear localization signal, and it helps in integrating the tDNA with the chromosomal DNA of plant. The first, which of the following is a fish cell line? Okay, the first permanent cell line was isolated from a fish. It was derived from the gonads of rainbow trout. Okay, RT designated rainbow trout and G for a scientific name. Okay, this is a scientific name of rainbow trout. The correct answer is RTGE. Okay, if you take some other cell line like Vero cell line and CV2 cell line and the COS1 cell line are derived from the kidney of African green monkey, and this LLCMK is from kidney of rhesus monkey, and this ST is from testis and fetal tissue of pig, and MAC is from alveolar macrophages of pig, and BHK21 is isolated from kidney of baby hamster, and CHO is from ovary of Chinese hamster, that is Chinese hamster ovary cell line. HeLa is from cervic carcinoma patient, isolated from that particular patient, and SHXY5Y is from neuroblastoma tissue of human, and L929 is a mouse cell line isolated from connective tissue, and A9 is a mouse cell line which is isolated from connective tissue of mouse, and this 3T6 is derived from an embryo of mouse, and NP3 is isolated from myeloma of mouse, and MDCK is from kidney of canine, that is cats, and SF9 is isolated from derived from ovary of fall armyworm. And if you take this HepG2, so this HepG2 is isolated from a liver tissue of a patient with hepatocellular carcinoma. Okay, it is a human hepatosoma cell line which is generally used for drug metabolism and hepatotoxicity states. The last question is, Biostil is a trademark name for high strength based fiber material which was made from recombinant spider silk-like protein extracted from a milk of transgenic. It was extracted from the milk of transgenic goat. The goat is called a spider goat. Though you all know spider has a capacity to produce a web, and it also has a capacity to produce silk-like protein. And the silk-like protein produced by spider have a high tensile strength when you compare with the silk that was produced by Bombyx mori. So what scientists has done is that initially they had isolated the silk and they had incorporated into E. coli and some other thing, but they didn't find success. But later on, they had isolated a silk coding gene from the spider and they had incorporated into a goat, just like a dolly, which was a sheep that was cloned first, and then they had the goat that has a capacity to produce milk, and in that milk you can able to find the silk protein. Okay, so what they will be doing means they will be extracting the milk from the spider growth or a genetically engineering transgenic goat. They will be purifying them, they will be removing the fat molecules, and then they will be precipitating out the silk-like protein by salting out process, and they will be converting each protein molecule into nanofiber, and this nanofiber will be converting into microfiber. Later these fibers are employed as the artificial ligament or fluid ligament because it is a good biomaterial. Okay, so it is extracted from the milk transgenic goat. Okay, so thank you friends, thanks for watching this video.